I'm Jane Bayani, and I'm a Principal Research Scientist and the Co-Director of Diagnostic Development at the Ontario Institute for Cancer Research. We often get asked by our patients, what happens when I consent to have my tissue donated for future research? In this video, we're going to show you a little bit of the work that goes on in our lab at Diagnostic Development. When the sample arrives, the information gets entered into a database. We ensure that it's been given an anonymized study ID number and the associated information required for the study has also been provided. The most common sample type that enters the lab are tumor samples that have been preserved and embedded into a block of wax. Before we can begin the proposed experiments, we need to make sure that the sample contains sufficient tumor material for the project. To do this, we need to visualize the tissue by shaving off a very thin section of the tissue and transferring it to a microscope slide. To visualize the tissue and the tissue structure, the tissue is stained with two dyes. We can then visualize the tissue either directly by the microscope or by taking images of the slides and visualizing them on a screen. Once we've identified where the tumor cells are and how much there is, we can begin the experiments associated with the project. Projects may focus on the location of the tumor cells as it relates to the surrounding normal cells that make up the cancer as a whole. One technology we have in diagnostic development allows us to use multiple fluorescence tags to look at different proteins or RNA molecules produced by the tumor cells and the surrounding normal cells. The location of tumor cells related to other cells in the cancer is important as we know that they influence each other. We can start to develop these cancer neighborhood maps and understand the extent of communication between cancerous and non-cancerous cells. For projects that require the extraction of just the tumor cells, we scrape off the regions containing the tumor and extract the DNA and the RNA from these tissues. Once we've extracted enough DNA and RNA, we can use this material to look for the key genetic changes using technologies like the next generation sequencing equipment we have here in diagnostic development and in other laboratories at the OICR. Diagnostic development is made up of an amazing team of highly trained technologists, research associates, computational biologists, and we work together with other researchers and healthcare professionals and answer the questions that are most important to patients and their physicians.